Translocations. The transfer of the genetic material from one chromosome to another chromosome is called as translocation. Translocation can be reciprocal as well as Robertsonian translocation. Based on the genetic material, translocation can be balanced and imbalanced. If there is no loss of the genetic material, then it is called as balanced, and if there is loss of the genetic material, then it is called as unbalanced translocation. Reciprocal translocation is a translocation in which the chromosomes they change the fragments between one chromosome to another chromosome or between the fragments of the same chromosome. Reciprocal translocation in homologous chromosome. In case of homologous chromosome, they exchange the part or the fragment of the chromosome with another chromosome. Reciprocal translocation before and after. In case of when there is no translocation, we can see that chromosome 20 and chromosome 4. After reciprocal translocation, the fragment of chromosome 20 will attach to the chromosome 4 and the part of chromosome 4 will attach to chromosome 20. So this is vice versa, reciprocal between two chromosomes. Here the example is of chromosome 4 and chromosome 20. When chromosome 4 and chromosome 20, they exchange the DNA fragments or the part of the chromosomes, such a translocation is called as reciprocal translocation. In case of Robertsonian translocation, that usually occur between two acrocentric chromosomes. During this kind of translocation, the smaller parts of the chromosome they are deleted and the larger fragments of the chromosome they fuse with each other and form a single chromosome. The larger fragments during this type of the translocation they fuse with each other and form a big chromosome. Robertsonian translocation occur between chromosome 13 and chromosome 14. This is a normal phenomena in the humans where Robertsonian translocation occurs, and this is usually seen that approximately one birth out of one newborns have the Robertsonian translocation. Robertsonian translocation with the help of diagrams, here we can see that there are two acrocentric chromosomes. This is acrocentric chromosome 1, this is acrocentric chromosome 2. As a result of the translocation or the fusion, they join with each other and form one single chromosome. So this is the result of the Robertsonian translocation. Or in other diagram, we can see that these small fragments of the DNA, they are deleted, and as a translocation, the large fragments, they join with each other. This phenomena is called as Robertsonian translocation. Types of the translocation based on the centromere or based on the non-reciprocal and reciprocal translocation. In case of non-reciprocal intrachromosome translocation, non-reciprocal intrachromosome, the change of the fragments within the single chromosome. Here, originally, this fragment was present on the, let's say, P arm. This is P arm and this is Q arm. Originally, this fragment was present on the P arm. As a result of translocation, it has joined on the Q arm. In case of non-reciprocal interchromosomal translocation, this type of the translocation is between two chromosomes. This is chromosome 1, this is chromosome 2. This fragment from one chromosome is deleted and it is joined on the second chromosome, that was chromosome 2 and 1. So B and C, which are the fragments, they are deleted from chromosome 1 and they joined with chromosome 2 here. The third type of the translocation is reciprocal interchromosomal translocation. In this type of the translocation, the fragment of one chromosome is deleted and it is joined on another chromosome. So it was deleted from chromosome 1 and it joined to a second chromosome. So these are the different types of the translocations. Many human diseases are caused by the translocations. Most common human diseases, they are cancer, infertility, Down syndrome, leukemia, and the abnormalities of the X chromosome are due to the translocations.